Hi, my name is Dr. Vasu K. Brown, and I am going to talk about Gene SNP DNA analysis today. And the lecture is done as an in-service on behalf of Alden Court in Massachusetts. They are a rehabilitation nursing home. And this is for the staff who wants to learn about Gene SNP DNA analysis. And the idea behind this is to learn your body language so you would be able to reset your body to its original health. The FDA disclaimer, this is to state that the contents are not evaluated by Food and Drug Administration. This product is not intended to diagnose, treat, cure, or prevent any disease. The purpose of this lecture is to educate healthcare professionals, so they can make informed decisions when it comes to themselves and their patients. Nutrigenomics is the new field of nutritional genetics and epigenetics. Based on the latest science from discovery of entire human genome, health professionals now can apply new knowledge to offer customized nutritional recommendations to individuals based on genetic variation and lifestyle. And the Nutrigenomics is the study of how nutrition through an individual's diet can affect his or her DNA, specifically in the way his or her genes are expressed. The way we inherit our genes and the lifestyle we choose and the environmental factors can affect our genes. But the idea behind testing is once you test it, it creates a blueprint for your life because the altered genes is only a once-in-a-lifetime testing. You do not need to retest it again. And this is a wonderful field for health professionals to get involved in because the mystery of nutritional genetics is being clarified so that you can communicate to client specific nutrition and nutraceutical choices based on their nutritional genetics. To understand nutrigenomics, we know that we take supplements, we take nutrition for specific illnesses or for weight loss. We do alter our diet and lifestyle, but still we do not get the response we need. So if you do not understand whether your genes respond to a certain modification, then you are using efforts that is not going to produce results. So a statistical genetic risk to a disease or condition does not mean that you will express the disease or condition. It is an important starting point. But then the freedom and responsibility is to choose your way, to choose the right diet and optimize lifestyle based on your genetic predisposition. This slide was taken from Professor Michael Muller, who is the director of Netherlands Nutrigenomics Center. The nutrigenomic field is rapidly growing, and FDA now has a division of personalized medicine, and also Brigham and Women has a department of nutritional genomics and preventive health department and a personalized medicine department. So many effort is placed, many different universities and educational institutions are seeking to implement research strategies that account for genetic, environmental, and cultural diversity that influence expression of genetic makeup. And the earlier we get involved, we would be able to offer choices based on our unique genetic predisposition. Nutrigenomics in life, there was research done on agouti mouse. They found there was two different mouse used for the research purpose, and the one mouse was given the regular diet, which did not have any nutritional supplement. The other one was given a diet which enhances the methylation via supplementing choline, betaine, and folate, which is B vitamins. And they found the way they could identify which was the healthier mouse was the coat of the obese mouse remained yellow, whereas the mouse which was given methylation turned darker with a browner skin, and also it maintained its slimness. And they tested both for the offspring and the second generation, and the mouse that was the mother mouse, which was fed the vitamins and nutrients, they maintained their well-being for 2 or 3 generations. So you can alter the offspring and also the future generations' health by modifying the mother's diet before, during, and after pregnancy. And through diet, you are switching on and off certain genes through protein expression of those genes, preventing the disease formation in the person or the research beings that's being tested, and plus the future generations. These changes to turn on and off gene expression through messenger proteins can be passed along to future generations, as I mentioned earlier. And the converse is also true. That is, the genetic makeup can dictate how we respond to certain nutrients. Let's say, even if you're getting the best nutrient and vitamins in the world, if in case your gene is altered, so even if you take the most expensive products in the world, you are not going to respond to the way like another person would. So it's better to customize your nutrition based on your genetic makeup. So the big picture is research should be driven by need to understand the origin of diseases, taking into account the totality of factors that may be involved, including the genetics, lifestyle, smoking, nutrition, physical activity. And there is a link between genes and health. Genes, through the proteins they encode determine how efficiently we metabolize food, how effectively we detoxify harmful substance, how we respond to certain conditions, among other functions. It is now known that there are many genetic variation in human population. When a gene contains a variation, a change from the reference or native state, the protein encountered by the gene may be altered. The genetic alteration or adaptive or beneficial, the genetic alteration can be adaptive, beneficial, or it could become maladaptive and you can express a disease process. Such genetic variation that result from one nucleotide change in DNA sequence is referred to as single nucleotide polymorphism. I spoke about this in the previous slide. So this is a statement made by Jeff Bland from Metaproteomics Lab, and their website is metaproteomicslabs.com. And it says it is now accepted that the person's state of health arises from dynamic interaction of environmental factors with her or his genetic uniqueness. These factors can positively or negatively impact the extent to which people will realize their genetic potential. And it has been demonstrated that nutritional factors have a major influence on healthy gene expression, which is genomics, protein synthesis, that is proteomics, and metabolism, metabolomics. As a result, a new patient-centered healthcare is emerging that will rely upon nutrition and other modifiable lifestyle factors to help people achieve optimal genetic expression. There are 4 base pairs in nucleic acid for DNA, which are adenine, guanine, thymine, and cytosine. These 4 DNA bases can be altered and can be tested for alteration, and those are tested by doing a genetic SNP, which is single nucleotide polymorphism, in 20 seconds. And this would give you a blueprint for your life of what you are susceptible to and how to avoid it. Learn what messages your gene are giving your body. Learn how you can, or learn what you can do about it. And the Gene SNP evaluates total of 30 genes, which goes over 41 SNP, single nucleotide polymorphism, which spans 7 different disease processes in the body. So what is Gene SNP? This is the future of optimum health. Through Gene SNP DNA screening analysis, your health and lifestyle considerations can be addressed by examining your unique genetic makeup. No longer you will have to accept one-size-fits-all vitamin and nutrition regimen, which is done for everybody. Instead, you can customize your intake of vitamins and nutrients based on your genetic makeup. Modern genetic research has made available to you a customized nutritional program and lifestyle recommendation. How does the Gene SNP work? Through the sequencing of our human genome, we find out what is altered. Our body, 99% we have similar DNA makeup, but 1% of this is unique to our genetic makeup due to our lifestyle and our hereditary and the life choices we make. And scientists have been able to visualize single nucleotide polymorphism, which are altered in our genetic makeup that can lead to pathology. How? Through SNPs, the gene codes protein to do the work in the body incorrectly if in case it is lacking certain nutrients or it exposed to more toxins. By understanding and replacing these nutrients, we create a new signal which are called epigenomes, which are able to reset the DNA and the genetics to its original state, so we lead a healthier life and not expressing these disease processes. So how does Gene SNP work? What happens when gene coding of protein is not correct? Certain biological pathway in our bodies can become damaged or less efficient over time. Example, there is glutathione S transferase theta 1 is an enzyme that in humans is encoded by GSTT1 gene. And this is a biological marker for biochemical detoxification. So this is called phase 1 detoxification. The enzymes involved at this stage are known as activators. They activate the substance that need to be removed, allowing the next phase to proceed. So they work by attaching a single oxygen or nitrogen atom to the toxic compound. Many of these enzymes are cytochrome P450, which are mixed function oxidases. So then the phase 2 detoxification, which is the second stage, which is called excretors. At this time, the excretion of the toxin takes place. For this, the enzyme need to bind the compound glutathione to the toxins activated in phase 1. So that's why when you do detox protocol, if you remember, many of you feel queasy, sick after you go through the detox process. It's because the phase 2 detox is where people get stuck. So usually I've seen many people do the detox for 1 or 2 days. First day they feel great, and then they start having headache, nausea, bloating, and not able to tolerate the detox. That's because they are stuck in the phase 2 part of the detoxification. There are, in the phase 2 genes, in that family, there are 3 different genetic expression of them available: GSTM1, GSTT1, GSTP1. And if there is a SNP encoding of this enzyme, then the variation can affect the rate at which toxins are removed from the body, and people who are stuck in this phase would not be able to tolerate the normal way of detoxification. So we need to modify their detoxification. The way I do that in my patients is